we have two speakers today, and we're going to hold a joint question and answer for them afterwards. And for our Zoom audience, you have a few ways to ask a question, both via chat and also just raising your hand, and Peter will take note of that. And so now I'm really delighted to introduce today's speakers, and I'm going to give you both bios together. So our first speaker is Sophie Breitbart, and Sophie is an urban ecologist and an evolutionary biologist. She's investigating how urbanization influences the monarch butterfly and its favorite botanical partner, especially common milkweed. And so after completing her BA in biology at Wesleyan University in 2016, Sophie worked as an environmental consultant, and she was assessing lake water quality and designing engineering plan sets for dam removals. She began her PhD in ecology and evolutionary biology at U of T in 2018. And in her free time, Sophie likes to instruct coding workshops and explore the incredible cultural and biological diversity within Toronto's neighborhoods. So I think you're going to be a member of TFN inevitably at some point. Our second speaker is Lucas Albano. And Lucas is in the final year of his PhD in Mark Johnson's lab at U of T, Mississauga. And his research focuses on how the surrounding environment affects adaptation in white clover across latitudes and continents. In his free time, Lucas enjoys hiking and watching professional cycling. So please welcome our two speakers and
Great. Hi, everyone. It's so great to see you all, and thank you very much for that wonderful introduction. My name is Sophie Breitbart. As we just learned, I'm a PhD student at the University of Toronto. I'm also in Mark Johnson's lab with Lucas, as well as Helen Wagner. You might also know me, if you're a really keen listener, I was on the Toronto Nature Now a couple years ago talking about scientific names, Latin names. But today I'm really going to be focusing on my research where I've been asking how city life impacts the ecology and evolution of common milkweed. So I wanted to start off with a question here. I'm just gonna hide this little guy. So I want to see a show of hands. How many of you think that a third, 1/2, or 2/3 of the human population lives in a city today? So how many of you think 1 out of every 3 people lives in a city? Okay, no one. What about 1/2? Okay, got a few hands. 2/3? Wow, all right. At least in person, 2/3 is the definite winner. Well, actually back in the 1930s, just around when TFN was founded, about 1/3 of people lived in cities. Actually, today, only just over than half of people around the world live in a city. So actually, I think we jumped the gun a little bit because 2/3 of people will live in a city not too far from now, around 2050, but we're not quite there yet. And so I think what this trend is showing us is that urbanization is this really booming process. It's not going anywhere. Actually, more than 82% of North Americans live in a city, so maybe that's why you all were thinking 2/3 is the right answer. But yeah, cities are really cropping up all over the world, changing our landscapes dramatically. And in addition to covering many parts of the world, these cities also tend to share some environmental characteristics. And so, for example, we know that cities tend to be warmer on average than more rural or natural areas surrounding them due to something called the urban heat island effect. Cities also tend to have lots of pollution, like road salt, smog, light pollution, noise pollution, the list really just goes on. And there's also a lot of habitat fragmentation in cities, which means that when you do find a green space, it tends to be smaller and more, isolated than what you might find in a more rural or natural areas with, bigger green spaces. And so if you're thinking, well, if cities tend to share a lot of environmental characteristics, does that also mean that the species that live in our cities share some characteristics? And the answer is yes.
Excuse me, I'm sorry to interrupt, but your slideshow is
when
not
we
working.
scientists talk about the ways that organisms look, we are talking about their phenotypes. And so a phenotype can be the patterning on a shell, it can be the color of a leaf, it can be your heart rate, anything that you can observe, pretty much. And so
Sophie?
we would say that urban populations tend to have different phenotypes than more rural populations.
Hello?
Sophie, can you
And so,
hear
for
us?
example, this group of scientists found that in multiple large North American cities, this plant called Virginia pepperweed grew to be larger than more rural plants, exhibiting phenotypic divergence. And they also found that there was a difference in these plants' abilities to produce offspring or reproductive success. So those same urban plants grew more seeds than the rural ones. And so in general, sometimes species can respond very similarly to these stressors associated.
The slides aren't moving on the Zoom.
Oh.
That's better.
Slides are in advance.
You can try to share a different window.
Yeah, you know what? It's in a different window, so we're going through the browser, so it's. Trying text. See.
So I should keep going?
Yeah, keep going.
Okay. All right, so
It's
thanks.
working. That's what-
Oh, great, it's working. Awesome.
Thank you.
So if you remember, as I was saying,
It's a good thing there's a digital team member on the Zoom.
Yes.
Get that.
Thank you. So as I was saying, sometimes species respond similarly to the stressors of urbanization, but sometimes they don't. And we think that there's so much variation in how different species respond to these stressors because, it's not just them responding to urbanization, it's their predators and how that affects them. It's their prey and how that affects them. It's their pollinators, their mutualistic partners. And so you can imagine that, for example, if we have a plant and that plant is, it really heavily relies on pollinators for its reproduction. If that plant's pollinators are struggling to find places to live in a city, if they're struggling to find food, then that plant might not be pollinated very well and its reproduction might suffer. But on the other hand, if its pollinators are having a great time living in that city, they love the heat, they can find places to live, and so on, then that plant's reproduction might be, doing just fine. And so that is really what I'm interested in, is not only thinking about how these plants are changing in response to urbanization, but also thinking about their interactions with their pollinators and other species. And so that really brings me to the first question that I asked in my PhD. How does the reproductive success of a native plant vary along an urbanization gradient? And of course, I'm not just thinking about the plant here, I'm also thinking about its pollinators, because we know that the responses might go hand in hand. And so when I'm talking about the community structure of these pollinators, I'm asking, when you go outside to St. Mike's and if you see a field of milkweed, which there are some milkweed growing in St. Mike's, I've put them on a Naturalist, and you see lots of wasps and bees and butterflies and moths, I'm asking, how many species do you see? How many individuals of each species do you see? Are there some species that are really abundant and some that are really rare, and that sort of thing? And of course, the plant that I'm using to answer this question is known as ninwish by the Ojibwe indigenous peoples. You might also know it as Asclepias syriaca or common milkweed. Just a few things to know about it. One is that it's a perennial, meaning it can live for many years, sometimes decades. It grows all over the place, urban areas, suburban areas, rural areas, natural areas. It really lives up to its name as a weed, and I love it for that. And it's also, of course, the host of the iconic butterfly, but it's also pollinated by at least 20 other insects. So I think it's really important in our ecosystems. And you can find it throughout much of North America, where it's considered a native plant. And specifically, I did my research in the GTA, the Greater Toronto Area. And so the first thing that I did was to draw a straight line from Toronto city center out towards Mississauga, out towards the Greenbelt. And I called this straight line a transect. And what I did was I found 80 sites with common milkweed within this transect, which happened to be drawn along an urbanization gradient. And so because of that, I used distance from the city center for each of my populations or sampling sites as a proxy for how urban it was. And then once I found a sampling site, I visited it twice per year for 2 years. First time in July and then in September. And so when I visited it in July, I wanted to know one thing. I wanted to get an estimate of male fitness by looking at how much pollen had been removed from these plants, trying to see, how well are they being pollinated. And with common milkweed, this is actually really easy because each flower has 5 pollen sacs or pollinaria, and you can identify them by seeing whether this little black dot that I've circled in red is present or if it's absent. So you can see that it's present on the left side and it's missing on the right side. It was probably taken by a pollinator, maybe like this bee that we can see here. And what happens is that they get these pollinaria tangled around their legs or other appendages, and eventually they either fall off or they deposit them into another flower. It's a really cool mechanism. So that was July. And then I went back again in September to look at a couple more metrics. So the first one, I wanted to get an idea of reproductive effort. How hard were these plants trying to reproduce by counting the number of peduncles per plant with each peduncle just signifying an inflorescence or a cluster of flowers. And secondly, I wanted to get an estimate of female fitness. And to do this, I counted the number of follicles or seed pods that oftentimes you see in the fall. They've broken open, they've split open, and all of those beautiful seeds are going to fly away. And then in 2019 only, a couple of very motivated undergrads went to these sites. They pulled out their binoculars. They sat about 3 meters away from these milkweed plants, and they just observed for 5 minutes which pollinators were visiting these plants. And so that is how we got our pollinator community structure data. And so what did I find? Well, before I show you my results, I'm just going to walk you through a typical figure. And so on the x-axis, which is the one going left to right on the bottom, we can see our metric is distance to the urban center. With very urban populations, they will be on the left side, and very rural ones will be on the right side. And on the y-axis, we've got our response variable. So here, it's the pollen area removed, or how well pollinated those plants seem to be. And so first, we'll start with the plant reproductive success measurements. So first we see that pollen removal was pretty low in the city compared to those more rural sites. Specifically, they were about twice as low in the city compared to the bucolic countryside, depending on the year. Twice or 4 times as much, 4 times as low. In terms of our other metrics, the number of follicles or seed pods, it really varied by year. Whereas fruit set, or the number of follicles per inflorescence, was actually higher in the city. But we didn't really see any differences in the number of inflorescences in general. And so now let's switch over to the pollinators. How did they do along that urbanization gradient? Well, we actually saw about 60% fewer pollinators in our urban milkweed plants compared to those more rural ones, 60%. But we actually saw more pollinator species in the city, but we didn't really see any differences in terms of species diversity. And so putting this all together, what does this mean? Well, it seems like for those urban plants, they experienced lower pollen removal, yet they still had pretty high fruit set. They had a higher number of follicles per inflorescence. And this is happening even though those urban pollinators, they're experiencing all sorts of things. There's no straightforward impact on those pollinators. And so to me, I think this is a really interesting finding, because usually we find that plants and pollinators are impacted by something like urbanization in the same way or in similar ways. And that's not exactly what we found here. And so to me, this is a surprising finding, and it's also pretty hopeful that maybe these species interactions are a little bit more resilient than we thought. Okay, so by now, I hope to have shown you another example of phenotypic divergence happening along this urbanization gradient. And so this is just one example of phenotypic divergence. We've also seen this in so many other species around the globe. But for the most part, we still don't really understand where this phenotypic divergence comes from. And so that's really where I wanted to head next. And so there are 3 main factors that can shape phenotype. I'm sure you know a few of these. So this first one is the environment. And the environment can shape an organism's phenotype through non-genetic means within a generation. And so, for example, if we've got a plant here and it's looking a little wilty and we decide to water it, and over a few hours we see it perk up, turn a little bit more green, its phenotype has changed. And that is likely due in large part to that environmental change of us watering the plant versus this other component associated with its genes, although those can certainly play a part. And when we talk about an organism's genes, we're talking about its genotype, just another word for that. And so, again, an organism's phenotype can be impacted by the specific sequences of DNA that it inherits from its parents and its grandparents, and so on. And finally, the third main factor that can shape phenotype is the interaction of these 2 factors, being a genotype by environment interaction, but we'll skip over that for now. And so again, we have seen so many examples of phenotypic divergence happening in cities, but we still don't really know, is it mostly due to the environment or genotype or a G by E interaction? We're really not too sure. And so that's where I wanted to take my second chapter of my PhD. I wanted to know, were those phenotypic differences that I observed earlier associated with reproductive success, but also many other traits, were those primarily due to evolutionary changes or instead just differences with the environment? And a little bit more generally, are the urban milkweed populations genetically different from the rural populations? And this question is it's a first step towards testing for adaptation, which is a very hot question these days. But in order to test for adaptation, I would first need to know, are these plants genetically different or not? And we thought, this could actually happen. We could actually see this because we have seen very quick and repeated instances of evolution happening in response to urbanization all over the world. And I think this is really something because for a long time, scientists thought that evolution took a really long time to occur, hundreds of thousands of years, millions of years, but more and more, and not just in the urban evolution category, but we are seeing evolution happening within our lifetimes sometimes. It can happen really, really fast. And so I was wondering, is that happening, with common milkweed? And so to answer this question, first thing that I did was I went back to those sites, and from as many as I could, I collected seeds from those common milkweed populations. Then I germinated those seeds. I ended up germinating about 1,000 of them and planting them in a common garden experiment. And I'm not going to cover what exactly a common garden experiment is in the interest of time, but it's really good for testing these sorts of questions, and you can ask me about it later if you'd like. But here's what it looks like. Again, so we took those 1,000 seedlings, we planted them in this common garden, and then for 4 years, I started measuring lots of phenotypic traits. And those traits belong to 3 main categories associated with plant defense, plant reproduction, and growth and development. And so what did I find? Did I find that these urban milkweed populations were genetically different from the rural ones? The answer is not really. And instead, it seems like that phenotypic divergence that I observed earlier, at least in those reproductive success traits, that seems like it was mostly consistent with environmental differences and not so much with genotypic differences. And so my next question was, wow, why not? We've seen genetic divergence in so many species in response to urbanization. So what makes common milkweed different, at least in Toronto's common milkweed populations? Well, I have a few ideas. So one is that maybe it has something to do with the life history traits of common milkweed. And so to step back, I'll remind you that common milkweed is a perennial, meaning it can live for many, many years, sometimes decades, and is also clonal. So it can reproduce sexually and asexually through these clonal offshoots. And so what this means is that if you were to find a field of common milkweed, it's possible that you could be looking at common milkweed plants that have been growing for decades, and there's actually just a few genetically unique individuals there. It could have just been a few individuals that were initially, they cropped up there and they spread lots of offshoots into the field. And there's not a whole lot of genetic diversity there. And so when you couple that with the idea that Toronto as a city is really young, it's less than 250 years old, maybe these plants just did not have enough time to be exposed to the pressures of urbanization and evolve. And maybe it's also because of the pollinators. So maybe it's happening that these urban pollinators are doing a really good job at going from urban plants to suburban plants to rural plants and just bringing this pollen all over the place, creating a lot of mixing of the pollen. And so in that case, oops. Oh, let's see, my screen went, it fell asleep. Well, in that case, that would prevent genetic divergence as well.
Just tap.
Tap
You
the
want it
screen.
to go full screen, just tap.
Thank you. And so these were just a couple of ideas that I had. But for my third and final chapter of my PhD, what I'm doing now is using some bioinformatic tools to try to answer this question by looking more deeply at the actual genetic sequences of these plants, of these rural and urban populations. All right, so as I said earlier, urbanization, it's this really fast-moving, intense global trend. It's really not going anywhere. More and more of us are moving to cities every day. You know, probably most of us live in a city. And I know that when I go outside of my tiny Toronto apartment, I want to get a breath of fresh air, and I want to see a thriving ecosystem in my neighborhood. And I think we need this sort of research to help us understand how our species are responding to the stressors of urbanization so that we can build cities that are more hospitable to everyone. And so I'd just like to take a moment here to thank you all for listening, thank my labs for all of their help with my research, as well as the invitation from TFN. This has really been a lovely experience. And I would be happy to take your questions after Lucas gives his own talk. So thanks very much.
Slideshow.
Yeah.
Right out.
All right. Hi, everyone. As Ellen said, I'm Lucas. I'm also a sixth-year PhD student in Mark Johnson's lab, and I'm very excited to be here talking with you today about the research that I've been doing over the past few years. As everyone in the room probably knows, phenomena like climate warming, species invasion, land use change, and urbanization are all human-caused impacts that are becoming more and more pervasive and are becoming more of a threat to global biodiversity. And understanding how species have adapted to these changes in the past or currently will help us understand how they might be adapting to future changes. One particular type of adaptation that my studies deal with is called local adaptation. And the hallmark of this local adaptation is essentially that populations have greater success when they're closer to their home environment than when they're moved further away from that environment. And success could be either growth or reproduction. So if there was an absence of local adaptation on a figure like this, it would look like a flat line. And if there was local adaptation, you would see this downward slope as these populations are moved further away from their home to which they are locally adapted. And a steeper downward trend would mean that the local adaptation in that environment is stronger. And the hallmark way of testing for local adaptation is through a common garden experiment, similar to the one that Sophie introduced earlier. I'll go into a little bit of detail about how exactly that works. So essentially what it means is you take populations from a whole bunch of locations and then you bring them all into one location and then you see how they do. In my case, I didn't use just one, I chose to use 4. So those were located in Mississauga, in Lafayette, Louisiana, in Montpellier, France, and in Uppsala, Sweden. And I have white clover populations that were sampled all across North America and all across Europe, identified by the orange and pink dots there. And I chose North America and Europe because Europe is the native range of white clover. And North America is one of its introduced range. So here it's considered to be an invasive species. And this is just the same white clover that you could find in pretty much any lawn anywhere. So that makes it very easy to go to all of these locations and sample all of these populations quickly and easily, and transplanting individuals into each of these 4 locations just to see how they do. So the goal of my experiment is to understand how variation in climate between all of those orange dots and all of those pink dots and those 4 common garden locations drives local adaptation in plant populations. And I can expand this goal out into 3 research questions. So first, are white clover populations locally adapted to the climate where they originated? And does the strength of local adaptation differ by the location where they're transplanted into? So Mississauga, Lafayette, Montpellier, or Uppsala. Second, how does local adaptation differ between the native range of Europe and the introduced range of North America? And third, are there spatial lags in adaptation to climate change detectable in populations of white clover? And I'll explain what a spatial lag is a little bit later. Now, in order to address these questions, we need some sort of measure of how climates are different from each other, or how different they are from each other. And we can do this by coming up with a measurement of climatic distance. So there are many, many ways to do this, but how I chose to do it was to extract climate data from every single location involved in the study, both where the populations were sampled and the common gardens. And the variables I chose were mean annual temperature, annual precipitation, the minimum temperature of the coldest month of the year, the maximum temperature of the warmest month of the year, and then precipitation in the driest and wettest months. And all 6 of those variables can then be combined into a 3-axis principal component analysis. I'm not going to explain what that is today, mostly because I don't know all the math that goes into generating this type of thing. But it works, is basically all you need to know. And what it can do is create a 3D plot that looks like this. So now all of those populations from the map, orange, which were in North America, pink, which were in Europe, and all 4 of those common gardens, which are shown in green, can now be plotted on a figure like this. And then I can calculate the distance on this figure between each of those points. So now I can calculate how different the climate from each of these population origins is from each of these common garden locations. So now I have my measure of climatic distance for every possible combination of transplanting one population from one location to another in the study. Now, this is the same figure that you saw earlier, except on the x-axis now environment has been replaced by climatic distance. So now, if you're closer to home, that's a climatic distance that's closer to 0. And if you're further away, that's a value of 4 or higher, however the sort of relationship played out on that 3D figure that you saw. And we're still looking for that downward trend as our indicator of local adaptation going on. So you're going to see a bunch of figures that look exactly like this, just with a bunch of actual data presented. So first, we're going to tackle question 1. Are white clover populations locally adapted to their climate of origin, and does the strength differ by location? So, like I said, we'll have climatic distance on the x-axis here, and our measure of success for this figure is the number of flower heads that the white clover plants produced. And overall, we do see that downward trend that was indicated on the previous slide. But it's mostly driven by steeper downward trends in Lafayette, that's shown in blue, and in Montpellier, that's shown in green. So the much hotter southern climates in Europe and in North America. Whereas in Mississauga and Uppsala, the more northern climates, we don't really see that strong of a signature of local adaptation. And if we look at the mass of all of the seeds produced by each individual plant, we see a very, very similar trend. So much steeper slopes for Lafayette and Montpellier, so the southern, more warmer climates, and more shallow slopes for Mississauga and Uppsala. So to answer our question, are white clover populations locally adapted? Overall, yes, but appears to be mainly driven by the more southern, warmer environments, so Lafayette and Montpellier. Next, how does local adaptation differ between the native and introduced ranges of white clover? We're just going to look at seed set mass here. And to start on the left, we're just going to be looking at the North American gardens. And the North American plants, as was shown earlier, are still in orange, and the European plants are still in pink. So here you can see that if we're looking at data just from the Mississauga Common Garden or just from the Lafayette Common Garden, only plants that were originating in North America show a signature of local adaptation. Whereas if we transplanted them over from Europe, it's essentially a flat line, so there's no adaptation going on there. Then if we look at the European Common Gardens, so Montpellier and Uppsala, we see the exact opposite. So now only the European plants are exhibiting a signature of local adaptation, and the North American plants are basically a flat line. So exactly the opposite trend between the two continents. So just to reiterate, in the North American Common Garden, so that's in the invasive range of the species, only populations that were from that invasive range exhibit local adaptation. And in the European Common Garden, so the native range, only those populations exhibit local adaptation. The populations transplanted back over from North America did not. And we can take this as evidence of very rapid local adaptation in the invasive range of North America in only about 500 years or even less since it was introduced over to North America from Europe. Now we'll move on to my last question about spatial lags in adaptation to climate change. So the natural question then is, what is a spatial lag in adaptation to climate change? So I'll explain that now. So just looking very briefly at the sampling design in North America here, if local adaptation is what we're expecting, we would expect to see peak success in plants that were transplanted the shortest distance, whether that's geographic distance or climatic distance. So we'll say that the orange dot that's just next to the second A of Mississauga is likely to be the best performing population of plants in the Mississauga Common Garden. And you would expect to see much lower success as you move further south and much lower success as you move further north. So if I was to plot a prediction of what would happen on a figure like this, so if I have how much the mean annual temperature changed between where the population came from and the Mississauga Common Garden location on the x-axis and success on the y-axis, local adaptation would look something like this. So we see peak success where the climate distance between the population origin and the common garden is the smallest. So essentially zero difference between that population next to the second A in Mississauga and the common garden would have the peak success. And much lower success when you move very far north in Ontario, and much lower success when you move all the way down towards Lafayette. So that would be a signature of local adaptation. If we were to see a spatial lag, you would see a shift in the peak of that distribution towards warmer locations. So you would see a shift south in where that peak success is. So now maybe populations from Ohio or somewhere around that area are now the most successful in the Mississauga Common Garden because they think they're at home. Whereas the Mississauga plants, now the climate has warmed to the point that they no longer are responding as well to what should be their home environment. And when we test for this, this is pretty much exactly what we see. So we'll look at a couple different measures of success on the y-axis here. So first, the proportion of the plants from each of those populations that survived. And you can see that the peak of the distribution has shifted towards warmer climates. So here, the peak is at about 3 or 4 degrees warmer. So now populations that came from somewhere that's 3 or 4 degrees warmer than Mississauga are now having the best success in Mississauga because they think they're in their home environment. If we're looking at the seed set mass, we see very, very similar trend. Again, there's a shift towards a shift of the peak towards a warmer environment. Here it's only about 2 degrees, but again, populations from about 2 degrees warmer than Mississauga now are having the most success. And if we look at the maximum plant area, so you'll know white clover as a ground crawler, so we can measure the total spread of the plant as it's growing. You see, again, about a 2 degree shift in that peak towards warmer climates. So for this particular study, there is evidence of spatial lags across multiple measures of plant success, so survival, seed set mass, and maximum plant area. And this is an indicator that the climate is warming more quickly than white clover populations are adapting. There is a caveat in that with my experimental design, I was only able to test this for the Mississauga Common Garden, so we're not sure exactly how this trend might translate to Lafayette, to Montpellier, or to Uppsala. So just to make some conclusions here, overall, white clover populations are locally adapted to their climate of origin, but it's strongest in more southern environments on both continents. We have evidence of rapid local adaptation within the introduced range of white clover in only about 500 years post-introduction. But no matter how fast it's adapting, it may not be able to adapt quickly enough to withstand the effects of climate warming. And why this is important is that, okay, we see that white clover can adapt very quickly, but the thousands and thousands of other species that are out there may not be able to adapt quite as quickly as white clover does, but white clover is still having trouble withstanding the effects of recent climate warming. So you can think about the consequences potentially for even slower adapting species that are out there. So the next question that you might be asking is, this study was conducted across continents and across latitude. How does this relate to the theme of today's lecture series, which is urban adaptation? And the short answer is that directly, it doesn't. But don't worry, because there was one organism that was involved in this experiment that did have to worry about adaptation in urban environments, and that was me. So to give you some context, all of my field experience previously was in places like this, or places like this, where if you saw another human being while you were doing research, you would be shocked. But for this particular white clover study, I had to move into environments that look a little bit like this. This is the front yard of a mega church in the US somewhere, or an environment like this, which is the backfield of an elementary school. Luckily, while I was sampling, school was out for the summer, so I didn't get too many people asking about what I was doing, but I would get some. And typically I would spend about an hour sampling in a site like this. And pretty much that entire hour I would be spent bent over like this, scanning the grass, looking for white clover seeds. And someone would come up to me naturally thinking that I was looking for something or I had lost something. And they would ask, oh, what are you looking for? Can I help you? And I would say, oh, I'm looking for white clover seeds. And they would say, oh, and then very promptly shuffle off in another direction. But overall, I would say the interactions while I was sampling were pretty positive. However, there was another issue involved with this particular experiment, is that it was planned to start in spring 2020. So you can imagine that might come along with some difficulties. So originally, the experiment was planned to be here in the beautiful Koffler Scientific Reserve, which is where Sophie's garden was located. But instead, due to COVID restrictions, it ended up here, which is right in front of Alumni House on UTM campus, which if you've ever been to UTM campus, you might recognize that it's basically the front lawn of UTM campus. So you can imagine that while I'm working out here for hours a day, I might come across people on their daily walks, and that was indeed the case. Quite a few times a day, I would say. Couples, families out on their daily walks would walk by and they would ask, "What are you up to?" And I would say all of these interactions were very, very positive and a very, very welcome break from squatting down and measuring plants all day. And my message to you is that I encourage you that if you are out naturalizing or walking in an urban environment and you see a scientist out there studying something, don't be afraid to ask them what they're up to, because it's a really good experience for us to be able to talk about our work with you. So yeah, don't be afraid to inquire, and we're always very, very happy to tell you what we're doing. So please feel free. I would just like to acknowledge my collaborators and co-authors on this project, along with all of the other research assistants and the current Eco Lab for all of their help with this work and their feedback on the presentation. And I'd also like to acknowledge one last change to the experiment that happened post-COVID, was that it was pretty difficult to be able to get people to come help me in the field. But my family gracefully volunteered. So my mom and dad, my brother and my sister and my aunt all spent quite a few hours in that Mississauga Common Garden helping me out. So I would just like to acknowledge their efforts as well. Somewhere between. Yeah, so that's all I have to say to you today. So Sophie will, I guess, join me up here, and we'll hold a joint question and answer session for that.
That was really
Thanks.
wonderful.
Both of you, amazing.
It's wonderful to get that back firsthand insight into science on the go. and if there are any questions that people have, we can start out. And let's keep it equal opportunity for both Sophia and Lucas. Yes, go ahead.
So thank you very much, both of you. They're really great talks. My question is for you, Sophie. So looking at milkweed, you basically found that grading between urban and rural didn't really show a phenotypic adaptation, right?
So
Can we try, because I believe the Zoom folks won't hear the question. If you could just try to repeat the question.
Sure. So I was asked if there was a phenotypic adaptation that I found between the urban and rural common milkweed populations. And so I'm just going to split up that question into two parts, because the first step was to test for phenotypic divergence. So just, do these plants look different? Do they behave differently? And then secondly, I wanted to know, because it seemed like we did find some phenotypic differences, are those phenotypic differences consistent with genetic divergence? So going a step further, and then if I had found genetic divergence, I would have asked, is this due to adaptation? And so I did not find genetic divergence. And so that means that I'd have a very low chance of finding adaptation to urban environments, at least in this, in common milkweed, in Toronto at this point in time. So it could definitely happen in the future, but we just didn't find it right now. So does that make sense?
It does. So my question is, have you considered the source of the urban milkweed? I, for example, have milkweed in my garden. It did not occur there naturally. I got it from, I think it was Lush. The Lush cosmetic store was having an open house, and they were giving away little milkweed plants, and I got a few of those, and I planted them, and they reproduced asexually. They put out so much. And then another garden I have at my cottage, I also have milkweed, and that came from seeds that I bought at some store in Toronto and threw out in this garden on an island in Big Rio Lake in Eastern Ontario. So for both of my milkweeds, I have no idea where they actually would have come from genetically and what the phenotype of their ancestors would have been. It's a really interesting question. Did you look at where those urban milkweeds may have come from?
So I'll try to summarize your question. You know, did we look at where these urban milkweed plants came from? Do we know whether they came from a nursery or if they were introduced into the city in some other way that was not, you know, as a result of like natural range expansion from the species? So one answer is we only sampled milkweed that seemed to appear as it had just grown naturally. So if we were sampling in someone's front lawn, which sometimes we did, we'd always ask for permission, but we would not sample the milkweed if the homeowner or the tenant had told us that they had planted it. So we did try to avoid sampling milkweed that had been brought in through like unnatural means, but at the same time, like we knew going into this that the Suzuki Foundation had this Got Milkweed program in 2018 or 2017, and yeah, it's great that people plant milkweed in their front yards, and so we did know that that could influence our results. And so actually what I'm looking at now is, again, these bioinformatic analyses. One of the questions that I asked was, how many genetic populations are there? And it seems like so far there's actually just one. So that might be a hint that there's just a ton of genetic variation in all of these urban milkweed plants, and we're not seeing, you know, specific clusters of like, these milkweed plants came from that nursery and these came from that one. But we were thinking about that in the beginning. So we're still not really sure where all of these plants came from. We can't distinguish if they got there naturally or if they were planted, but that's part of urban ecology and urban evolution is people bring stuff with them and they plant it, and then that, you know, flies away elsewhere. So it's part of the fun of it, I guess.
Okay,
Thanks.
thank you. Really interesting stuff.
Thank you.
Any other questions in the room? Nancy first, then Michelle.
Okay, I have a question for Lucas.
Sure.
And it's kind of parallel to your question, just has to do with the genetic origin of the North American clovers. They've been around for 500 years, but how many introduction events do you think could have occurred? And what would be the impact on your results?
Right, so the question was, how many introductions were likely to have occurred from Europe into North America, and how might that have impacted the results? The short answer is we don't know, but we expect that it's much more than one. So the best information that we have is that white clover was brought over in mixes of other seeds that were brought as agricultural crops. And you could imagine that a ship arriving in Nova Scotia might have had a different population of white clover from a ship that arrived in Florida. So there could have been many, many introductions sort of along that latitudinal transect from south to north in North America. And it's possible that depending on what populations were brought over and where, that accelerated how strong local adaptation occurred along that gradient within the last 500 years, or it could have slowed it down if the populations that were brought over were kind of the opposite of the climate that they were already adapted to back in Europe. So we don't really know, but there's plenty of potential scenarios that could have been involved in speeding up or slowing down local adaptation in the introduced range. Michelle?
I have a question about the common gardens. I'm curious where they are in the world and how they came to be, and particularly curious about what you both foresee given our rapid climate change and all the political changes, because it strikes me that these are a really important thing to have for your research. Sorry, that's kind of a big question, but can you talk about some of that? I hadn't heard that term before.
Right. So the question was where these common gardens are essentially, and you know how we use them. So I guess the answer to that is a common garden can be anywhere you want it to be. So there are some, across the world that scientists have maintained for 20 years, 50 years, like very, very long-term studies. For myself and for Sophie, we both installed our common gardens ourselves and removed them ourselves. So it kind of depends on the design of your study, how long-term you want to look at things. So there are some, throughout the world that are very, very long-term studies that, I couldn't point you to one particular location. Maybe Sophie knows, I don't know. But yeah, for us in particular, we sort of installed them ourselves in locations that were the best for what we wanted to study.
Yeah, I think that's a great answer. Yeah, so both of I guess my common garden was in Newmarket, Ontario, part of the Clover Scientific Reserve. And one more thing that I wanted to add is that you can also have common garden experiments that don't involve plants. You can raise lizards or other types of animals with the same sort of design, where you bring in whatever species you're studying in, and they come from all sorts of environments, and you raise them in the same environment, and then you see what they look like or their offspring looks like after being raised in that same environment. So it can actually be used for all sorts of species beyond plants.
Sure.
So I have a question for Sophie, too, and it had to do with the impacts of predators on your milkweeds. So I think you had reduced pollinators in an urban setting, close to zero, but you still had sort of the same level of seed set. And so I think you also presented these results, but I wanted to get clear. So there were fewer predators in the urban setting too, is that correct? Like you had a smaller percentage of weevil damage and that kind of thing.
So
And did that affect the higher seeds, relatively high seed set you had in an urban setting?
So the question is, did I find fewer predators in the urban environment, and did that impact the seed set, like the fruit set? I actually didn't look at the predators. Actually, some of my colleagues did do a study where they looked at milkweed herbivores. So herbivores are the insects that eat the milkweed, not exactly that eat the pollinators of the milkweed, but they found, it was very complex, their findings, so I can't actually remember what they found, and I can't, summarize it here. So we did not look at the predators.
I was thinking of just because monarch butterflies can potentially be pollinators, but they're also predatory herbivores.
Absolutely.
Too.
Yeah, it's a great question. I can, if you come up to me afterwards, I can try to point you towards that paper if you'd like, but yeah, I'd like to know too.
Another question for both of you to do with the common garden question. So each of you planted a common garden. From the sounds of it, the assumption that the plants growing in the common garden were growing in the same substrate under the same temperature and climatic conditions. Is that right? Did you test the soil in the common garden on several transects to ensure that assumption was correct?
Yeah, so the question was about the soil conditions in the common garden and how that might affect differences between where the populations were sampled and the common garden location. I would say that you've probably picked out one of the biggest criticisms of common garden experiments, particularly with plants, is that a lot of times it's either not feasible or too expensive to harvest soil at all of the locations and compare that to the common garden soil. So I would say it's a very understudied aspect of the type of work that we do, because most people don't account for it. And I don't believe you accounted for soil conditions either. I know that I didn't.
Well, yeah, it is. It can get complicated. Something that I did was I used a blocking design. So I split up my common garden into 4 quadrants, and I put that sort of variation into my statistical models to try to account for variation that was inevitable in that field. So like differences in the elevation of the field, because it's huge. It's a huge field. Differences in the soil, differences in all sorts of things like where the squirrels would like to dig and like throw out our plants and things like that. So yeah, that is how I try to account for that. But yeah, as Lucas said, it can get tricky. So it's a good thing that you pointed out there.
Yeah, it could be just, if you want to like really account for it, you would have to harvest soil from every single site that you sampled your plants at, and then at the common garden site as well, which is would be very difficult to do. And so usually it boils down to trying to make sure everything at the garden, we do everything that we can to keep things consistent, or like Sophie did a blocking design to kind of account for as much variation within the garden location that we can. But yeah, it's very difficult to account for how things might be adapting to their local soil conditions as opposed to the climate conditions or urbanization or something like that.
No more questions? Thank you very much. I don't see any questions online. And for some reason, the video stopped. So they can hear us but can't see us.
It's okay.
Thank you very much, both of you. End it for the folks online.
I don't think we have any more announcements really, and I'm really delighted that the two of you could present together and give us a range of the kind of work that's going on right now that these are the kinds of questions that maybe not in such a sophisticated way, or they come to mind as we wander the fields. I wonder about, and rest assured,